24 hours. After that, they'll be accessible on ACS Webinar's website. Finally, just a reminder about lab notes, which are an exclusive benefit to ACS members with the premium package that feature a downloadable 2-page summary of key concepts and essential terms from each ACS webinar. Today's moderator is Cassandra Kallman, an assistant professor and C. Pritt Scholar in Cancer Research in the Department of Chemistry at the University of Texas at Austin. Following her MS and PhD in chemistry from UC San Diego, she became an American Cancer Society's postdoctoral fellow at Northwestern University. Her research group at UT Austin focuses on developing glycol materials to address critical challenges in both human health and the environment. And with that, Cassie, I'll turn it over to you to get us started.
All right, thanks, Michael. Hi, everybody. I'm really excited to be here and to be the moderator of today's webinar, which is, of course, on the topic, Polymers at the Interface of Sustainability and Health, Degradable Design and Synthetic Insights, where today we have a specific focus on the role that carbohydrates play in this space. And so my role is I'll be introducing our speakers and fielding questions from the attendees. So please enter your questions in the Q&A as they come up. And after both speakers have presented, we will discuss them. And so without further ado, it is my distinct pleasure to introduce our first speaker, Dr. Kevin Edgar, who is a professor in the Department of Sustainable Biomaterials at Virginia Tech, as well as an associate dean in the graduate school. He is an active member of the Macromolecules Innovation Institute and the NSF Glycomet Materials Innovation Platform, and as well was a founding co-director of Virginia Tech's Infectious Disease Interdisciplinary Graduate Program. Dr. Edgar has received numerous awards for his outstanding mentorship, as well as research achievement from the Virginia Tech College of Natural Sciences and the Environment, and he was named as only one of 25 members in the Virginia Tech Academy of Faculty Leadership in 2023. In addition to his academic appointment, Dr. Edgar is an editor-in-chief of the journal Carbohydrate Polymers, as well as an editorial board member of both Biomacromolecules and Cellulose, where he was also a former editor. Dr. Edgar has also been a longstanding leader in the American Chemical Society, where he's chaired his local section, he's chaired the cell division, and he's served and is currently serving on multiple committees. Currently, he serves in ACS governance as a counselor, and he is an elected member and elected chair of the Committee on Nominations and Elections. And finally, just one quick note is that Dr. Edgar was also a founding organizer of the Gordon Research Conference on Polysaccharide Chemistry, which has created an innovative approach for rotating the conference around sites in North America, Europe, and Asia every other year. And so it is my pleasure once again to introduce him to you today. And Dr. Edgar, the floor is yours.
Thank you so much, Cassie. It's my pleasure to be here today. So we're going to talk today about sustainability. We're going to talk about polysaccharides. We're going to focus on blockopolymers and synthesis and regioselectivity. There's a section about hydrogels, which we may not get to. Well, it depends on how fast I go with the other sections. But let's get started. So you heard a lot about me. I don't need to hear too much more, but the theme of our lab is certainly to take renewable, abundant, diverse polysaccharides that nature gives us for free, as long as the sun is shining and there's CO2 in the air, and use them for better purposes. And so sustainable plastics certainly is going to be one thing that we talk about a lot here today. And why sustainable plastics? I think we all understand, but just it's sensible to underline that plastics, first of all, are very beneficial in a lot of ways, right? They're nearly unbreakable. I tell the story of having been a young man working in a grocery store and going around a corner with a load of drinks and glass bottles, some of which fell off and broke on the floor right in front of my floor manager, who thankfully did not fire me on the spot. The youngsters that are in the grocery stores today are dealing with drinks in plastic bottles, which are much less inclined to break. They're also lighter, so it takes less energy to move them around. So a lot of good things about plastics, but we have to worry about degradation, certainly. We have slow degradation of petroleum-based plastics, and they persist in the oceans. They form gyres in the oceans, and those can take a long time to go away. We worry about their impacts on wildlife, right? Six-pack rings can get caught around a fish, around the neck of a bird. We have birds like uni birds that happen to find plastics interesting and they eat them. and eventually their intestines get filled up with plastic such that there's no room for nutrients anymore, and they actually literally starve to death. And of course, we all are concerned about microplastics in the environment and the fact that they keep getting found everywhere. They're found in virtually every organ of the human body. We don't know how toxic they are, but they're there, and we just assume they weren't there. It's a reasonable question, and given the context of this lecture, as to whether biodegradables would help, because biodegradable plastics, of course, will also go through a microplastic stage. But I think we can confidently predict that if a microplastic is a biodegradable microplastic is biodegrading, that is going to go through that small piece stage much faster than a petroleum-based plastic that might take decades to degrade. And then, of course, the raw materials that we use to make plastics, generally speaking, are not renewable, and that's a concern as well. So how do we address the problem? Biodegradability certainly is part of the solution. I always like to point out that we're not the first people. The people that are publishing today, besides ourselves, are not the first people to think about this. If you go back even into the 1990s, there's some great work by Charles Buchanan at Eastman Chemical Company. He used to share a lab with him. He's one of the best polysaccharide chemists on earth. And at that time, if you look up in the upper right there, you see cigarette butts, right? So cigarette butts are made of cellulose acetate. People back in the day used to see a lot more of them discarded in streets and other places than you do today, since there's less smoking. But you would see those cigarette butts, and they would sit there, in a puddle. Today, they'd be there a week, they'd be there 2 weeks. People started to assume that they weren't biodegradable because they persisted apparently in nature for so long. And Charles was able to ask questions to address that issue. The way he did it was to make cellulose acetate that was radio labeled with C14 on the ester groups and expose that to a composting environment, to a degradation environment, and collected the CO2 that came off, which could only have come from biodegradation of the polymer, right? Carbon-14 CO2 comes from biodegradation. And he was able to show what you see in the graph here, which is a comparison of cellulose acetate that is very similar to that cellulose acetate that's in those cigarette butts. That's here, with a degree of substitution of 2.57. I'll pause and note that degree of substitution means the number of positions that are substituted per monosaccharide. The maximum for cellulose is 3, right? So 2.5 out of the 3 are substituted. That's the cellulose acetate that's in cigarette butts and screwdriver handles and tape and things like that. So it does biodegrade, and it biodegrades reasonably fast. Note the x-axis is in days. So over the course of a few weeks, it's reached maximum biodegradation. If you reduce the degree of substitution a little more, make it a little more hydrophilic polymer, it biodegrades even faster. And it turns out that's a pretty good plastic too. So we have biodegradable cellulosics, particularly for objects that are going to be more durable, that are going to hang around longer. And one of the questions, of course, is how they will compete versus other polymers on price. So one question that you might want to ask is, what about starch? Right? There's an awful lot of starch out there in nature. It biodegrades too. It's natural, it's renewable, it's biodegradable, and it has the advantage that it's a little bit easier to get from its natural sources than cellulose is because of some of its properties. It's got this one special property. So starch can be thermally extruded. I don't know of any other polysaccharide for which that's true. Starch has such affinity for water that it's plasticized by water. And water obviously is the ideal plasticizer, right? Benign, cheap. So we can plasticize starch with water. Once we plasticize it, we can thermally extrude it. And what you see here is an experiment in which they extruded a sheet of starch, they made a tray out of it, and they just put water in the middle of that tray. So we can say it's a great idea to use starch as a material, a raw material for plastics, because it's got all these great properties: biodegradable, abundant, renewable, and can be extruded. But then we might say, maybe not such a good idea, because you can see when you put the water in that starch tray, over the course of just about an hour and a half, the tray completely disintegrates. So that's a problem. One of the things you might want to use things like starch for is single-use plastics. Imagine the scenario in which a fast food restaurant All of the plastics, all the cutlery they use, the glasses they use, the trays, the dishes, everything is made of a biodegradable plastic, such that when things go in the garbage, it's food and it's biodegradable plastic. And that could go to a municipal composter, could be composted to make soil that can be used to grow crops. So there is no waste, right? You're turning your waste into a value-added product. You're able to do that because of the biodegradability of starch. Not so good though if you're sitting there having a nice lunch, your starch-based tray is sitting there and it's wet. You get up, you walk towards the garbage, and the tray falls apart and everything falls on the floor. That's not so good. So starch-based objects themselves are too water sensitive, so it's a bad idea. But it could be a good idea. So we can acetylate starch. We can make acetate esters of the starch hydroxy groups, and that reduces water sensitivity. And fortunately, once you do that, no matter what the degree of substitution is, even if it's fully substituted, it's still biodegradable and it's still extrudable in the presence of water. And in fact, that's what this little red curve is here. So starch acetate tray prepared by extrusion in the presence of water. And you see that if you put water on that tray, it doesn't disintegrate. It's going to last at least for hours, long enough to be a single-use plastic. So it could be a good idea. Yeah, but it's not such a good idea. So starch acetate, starch itself in nature is not a structural polymer. In nature, starch is used to store photosynthetic energy. That's what it's for. It's not for holding things up the way cellulose is, right? Cellulose being the steel I-beam of the plant world. Starch is not a good plastic. It's not a strong polymer. And starch acetate is really too brittle for plastic applications. So what can we do about that? We want to use renewable materials, especially for single-use plastics. We love that idea. We'd like to get there. How do we get there, given the properties of starch? What about blending? What about blending with something like an aliphatic polyester, polylactic acid, polycaprolactone, things like that, that are also renewable, that are also can be made from renewable materials, that are also biodegradable? Could be good because they are more hydrophobic, right? So they're going to be more resistant to water. They're more flexible. They've got all those methylene groups that allows them to absorb impact, so they can improve the impact resistance of the starch-based plastics. That's all good. One little problem is they're not miscible with starch esters. They don't mix well with starch esters. And if we have two polymers that don't mix very well, when we mix them, we're going to have a plastic that has large domains and there are boundaries within those domains, between those domains. And those boundaries are weak points in the plastic. So that's a problem. There are objects out there that are made out of PLA and starch acetate. Those blends are commercial. You have some cutlery that's made that way. You have some shampoo bottles that are made that way. And what you find is a couple of things. First of all, none of those objects are transparent because immiscible blends are not transparent, right? So you can't make a transparent material. Should you want to have one of those nice transparent shampoo bottles, can't do it with this particular blend. Also, you'll notice the shampoo bottles that you get are always the ones that are in the fancy hotels where they're telling you it's a green shampoo bottle, right? It's not made out of polyethylene, not made out of a petroleum-based plastic. It's made out of renewable plastic, but they're always small. They're always 2 ounces because 2-ounce bottles are lightweight, and if you drop them, they don't break. The bigger bottles, though, you might want to have at home in your shower. Nah, you can't make those out of starch and PLA because the impact resistance is not high enough. Those would break. So how do we get around that? And I'm going to talk quite a bit about the work of my former student, just graduated, Jeffrey Thompson, who's a wonderful grad student. He did this work. So if we think about starch, starch is not one polysaccharide, it's actually 2. It's a linear polymer of glucose called amylose that's linked. We call this an alpha 1,4 linkage. And then it's another polymer that has linear amylose portions, but also has branches, also has glucose branches, and actually generation upon generation of branches. It's actually a natural dendrimer, and that's called amylopectin. So that starch polymer can be converted into starch acetate, right? And we've talked about that. It can also be enzymatically converted to glucose, which can be enzymatically converted to lactic acid, which can be made into lactic acid dimer that can then be polymerized to make polylactic acid. So polylactic acid also comes from starch. It's also a renewable based material. right? So that's great. We have two renewable materials and we just have to mix them together. But like I said, they're not miscible. And you can see that in this photomicrograph here where the domains of the starch, of the polylactic acid and the starch acetate are very large, right? They're just not blending with each other. And that's going to lead to poor physical properties. So what we wanted to do was to come up with a compatibilizer, something that could sit in between the two phases, reduce the energy between the two phases, and then therefore make the phases smaller and smaller and smaller, approaching miscibility. And so how do you do that? The thought that we had was to take an amylose-based polymer and to put branches on it made of polylactic acid. And what kind of architecture do you want, right? Do you want to have a lot of branches? Do you want to have short branches? Do you want to have long branches? Thought about that at the beginning, created a hypothesis. We really did. This isn't one of these hypotheses that people put out after the fact. This really was our starting hypothesis, that what we wanted was relatively few polylactic acid grafts on the amylose chain, but long ones, long, infrequent grafts. And the thought was that the amylose would lay along the surface of the starch acetate, and the long polylactic acid, infrequent polylactic acid chains would penetrate into the PLA domains, thereby reducing the energy, thereby increasing miscibility. How to do that? Well, to do it, we wanted to make derivatives, we wanted to make copolymers of PLA and amylose. We wanted to do that very selectively, regioselectively. What do we mean by regioselectively? Regioselectively means you know where the branches are. They're attached at certain hydroxyl groups and they're not attached at other hydroxyl groups. I always say, if we want to understand the structure property relationship of a material to make it do what we want it to do, it's hard to understand the structure property relationship if you don't really understand the structure, right? And this is an issue in polysaccharide chemistry. It's hard to make things selectively. And so there's this nice work in literature to make regioselectively substituted polysaccharides, but it's hard to do. Their literature examples are rare, and it's because polysaccharides are big molecules that diffuse slowly. They have lots of poorly reactive nucleophiles, hydroxy groups, and other nucleophiles. They're poorly reactive because of that slow diffusion, but also because of the fact that the approach angles are quite limited on a polysaccharide. Because the reactivity is low, to modify them, we have to have harsh reaction conditions, temperature, catalyst, things like that. It's really hard to make a reaction selective when you're carrying it out under harsh reaction conditions. Well, one of the reactions that we thought would be useful is this bromination reaction. So it's brominating amylose selectively in the C6 position. And the most regioselective reaction I know of in polysaccharide chemistry was invented by Furuhata back in the 90s. And that's an N-bromosuccinimide bromination of the primary alcohol groups on a polysaccharide in the presence of triphenylphosphine. And so what Jeffrey needed to do was to brominate at C6 selectively and then acylate all the other positions. And the beautiful thing of this chemistry is you can control stoichiometrically how many bromines you're putting on at C6. And that's going to be the place where we attach the polylactic acids, right? So if you want to control that frequency, you have to control the degree of substitution of bromines. And by carrying out this reaction this way, brominating first, then acylating all the other positions, that allows you to control the degree of substitution of those bromines completely, as well as where it's substituted. And we can see by proton NMR where we have 3 acetyl signals. C is the signal for the C6 acetate. So the more it's brominated, the weaker that signal gets. And we can see that we can get materials that have an entire range of substitution. So now I'm showing you the polymer as its azide. The substitution of that C6 bromine by sodium azide is quantitative. All right, so now we have the azide at C6, and that's going to be what we're going to click on the PLA side chain with. And you can see that that goes from a degree of substitution of 0.06 all the way up to full substitution at C6. Full substitution at C6 would be DS1. And we're going to test out the click reaction that we want to use, copper catalyzed click reaction, by using T-butyl propargyl ether, which has got this very nice reporter T-butyl group. It can tell us what's the degree of substitution of the group after the click reaction. And we can see that T-butyl group and very clearly in proton NMR. We can use that to quantify how many of these clicks have happened. And what we can see is it's essentially quantitative, actually all the way from bromide to azide to click product with the triazole ring. This X over here is also important. The X is telling you how many unsubstituted monomers, how many monomers that don't have this triazole ring do you have in between branches, right? So we can go anywhere from 0, so we can have a branch at every monosaccharide, all the way up to close to 20. We can have close to 20 unsubstituted materials, unsubstituted rings in between the branches. So we're controlling that branch frequency. What about the length of the branch? We said that was the other part of the hypothesis. We want to control the length of the branch. And how are we going to make a PLA derivative that we can click on? The way we're going to do that is to react lactide with propargyl alcohol. So use propargyl alcohol as the initiator, ring opening polymerization, use a catalyst that in our hands in this system really worked well to control dispersity. So you see dispersity is very low, right? That means that all the chains essentially are the same length. We can control thereby the chain length, right? By stoichiometry, we control the chain length. And that means we can make materials that are a little bit below and a little bit above the target length. And we can see here by this particular value, that tells us that the initiation was almost entirely by propargyl alcohol and not by water, which means Jeffrey did a good job of controlling the water content of this reaction. So we have it, right? We have all these PLA chains that are alkyne terminated, so they can participate in the click reaction, and they have a controlled length. So we have controlled lengths, we have controlled degree of substitution of the click sites, right? And we have controlled position of those click sites. So we've accomplished what we were trying to accomplish. Then there's this one remaining little issue of, can we get two polymers to click together, right? So this is not the easiest system in the world, but it turns out that it works. Copper catalyzed click reaction of the PLA terminated with the alkyne, and the amylose with the azide groups at C6 works very well. Very high yield, very high conversion. All of our spectroscopy, all of our DOSY experimentation showed us that they were in fact attached to each other and that we had the proper material. Right, so we've done it. We've controlled everything that is controllable. The one thing we didn't control and couldn't control is sequence of the monosaccharides, but every other dimension of structure is controlled. Okay, that's nice, but does it work? Good question. So here we're going to look at optical spectroscopy that looks at the phase separation between starch acetate and PLA. Right, so this is the so-called base blend. We're taking starch acetate, we're just taking PLA, we're just mixing them together. I told you already they're not miscible. You can see that here in this slide. You can see the blue domains of the one and the green domains of the other entirely separated, and there are large domains as well. What about a material that's got 100% graft density? What that means is it's got a PLA side chain on every monosaccharide, right? Remember from previous slides, we made that. And we said we didn't think that was going to work very well, and it doesn't work very well, right? It does compatibilize a little bit. The domains get a little bit smaller, right? So some positive signs that kind of structure could be a compatibilizer, but 100% graft density is just too high. So is 1 and 3, right? So if you have one branch for every 3 monosaccharides, it's better. It gets better than 100%, but it's still got lots of domains and relatively large domains of the 2 polymers. What if we go down to about 1 branch for every 25 monosaccharides? So in that experiment, what we saw was that we couldn't see it optically. We couldn't see separate domains optically. I wouldn't claim that this is a fully miscible polymer blend. It's probably not, but by optical spectroscopy, it is. We can't see the separation between the domains. So controlled length of the PLA, controlled graft density, does in fact, according to our original hypothesis, bring us to a block copolymer that is an excellent compatibilizer between starch acetate and PLA. So we go from 2 polymers that are both renewable and degradable that were immiscible and that could not make a good plastic to materials that in fact are miscible and now can make a good plastic. Let me switch to another topic. And I'd like to talk about cellulose as a bioplastic. What about cellulose as a bioplastic? Well, processing of cellulose is always difficult because cellulose is not soluble in any single solvent and it can't be extruded. You can't observe at glass or a melting temperature with cellulose. So you have to sterify it. And you can make esters like acetate and propionate and butyrate. Those are commercial materials, have been for a long time, and they can be used to make plastics. But to be used to make plastics, they need plasticizers. They need low molecular weight plasticizers. And the fact that you have to use a plasticizer that can be come out either by volatilization or by coming out into if you're using it as a container for some liquid materials, right, it could extract out. That can negate the perception of low toxicity, biodegradable, sustainable. That low molecular weight plasticizer is harmful to applications. Still is used in applications, used to make eyeglass frames and toys and toothbrushes. To broaden those uses, though, to broaden to more durable plastics that would last longer but would still be biodegradable at end of life, what would you have to do? Well, you want to make it self-plasticizing so it doesn't require those low molecular weight plasticizers. But long chain esters work for that, but long chain esters biodegrade slowly. So can we get the perfect mix of longer chain esters and shorter chain esters to hit a biodegradation rate that we'd like to hit? We can have some mechanical issues like creep with some of those short chain esters. And of course, cost is an issue. They have to compete on cost with petroleum-based plastics. So can we make cellulose esters that work as well as plastics, as well as petroleum-based materials that are in fact biodegradable and can be used for more durable objects? One of the ways we'd like to approach that is through, again, regioselective modification. And so in Germany, Iwata in Japan, developed a methodology for protecting cellulose with methoxytrityl groups at C6, and then subsequently acylating to put acetyl groups or other ester groups selectively only on C2 and C3, taking that trityl group off under acidic conditions, and then esterifying at C6 with a different ester group to make material that largely is substituted exactly where you want it to be. In this case, acetyl groups at C2 and C3, propionyl group at C6. That sequence that they ran required 4 different isolations, right? So it was a way to make materials that were interesting to study, but it was not at all a practical way to make materials. And so we had a hypothesis. My student Jeffrey in particular had this hypothesis that esterification of cellulose can occur under both acidic conditions and basic conditions. Well, putting on that trityl group requires basic conditions, and taking it off requires acidic conditions. So we had this hypothesis that we can combine putting the trityl group on, acylating at C2 and C3, both under basic conditions, isolate one time, and then under acidic conditions, both take the trityl group off and acyl with a different anhydride at C6. So that was the hypothesis. Would it work? So what Jeffrey found was that methoxytrityl group, in fact, could go on and dimethylacetamide lithium chloride solution under basic conditions. Acetylation could occur under those same basic conditions. And that gives us fully substituted products that have the amount of trityl group at C6 that we want. We can control that completely. Because this product is fully substituted, it's nice and soluble. It's all set up to do the chemistry as the next 2 steps. If you tritylate cellulose, you have to tritylate it completely to get a soluble material. If all you're putting on is that trityl group, you have to tritylate it completely to get a soluble material. Partially tritylated materials aren't soluble and therefore can't be carried on into further reactions. So Jeffrey's chemistry was ideal in that it was fully substituting it. If you chose to only put trityl groups on every 10 monosaccharides, that's fine because the other 9 are going to get acetylated, right? So that worked beautifully. And then under acidic conditions, what he found was trifluoroacetic acid was the ideal acid You could take off the trityl group and then acylate in the same pot, in this case with propionic anhydride. So getting the same kind of product that you saw in the previous chemistry, except for one thing with 2 isolations instead of 4, much more economical, much faster, much higher overall yield, same selectivity, same Regia selectivity as the other process, and also with complete control over the degree of substitution at C6. So you can make whatever material you want to by this chemistry, which you cannot do by the other chemistry. Also, what we saw was there was essentially no loss of degree of polymerization. In other words, the polymer is not getting chewed up by these conditions. It's remaining, the size of it is remaining intact through these 4 steps and 2 isolations. So it gives excellent control over that degree of substitution at C6, as I alluded to, everywhere from 0.01 degree of substitution. So essentially, you could, if you think of it as creating branches, 1 branch for every 100 monosaccharides to a branch at every monosaccharide and everything in between. Also, the nature of this chemistry is such that you can attach ester groups at C6 that you could not do by conventional chemistry. So you can make some of these longer chain esters. You could also make branch esters like isobutyryl that are very difficult to make by other chemistries. What about acid chlorides instead of anhydrides? And we thought, well, for that last step, acid chlorides might work and might not require an added acid, because anybody who's ever opened a jar of an acid chloride understands that every acid chloride's got a little HCl in it, right? So we add the acid chloride, the HCl that's already present in the acid chloride catalyzes the removal of that trityl group and catalyzes acylation at C6. So we're able to make hexanoates at C6, for example, by this chemistry. We're also able to make isobutyrates by this chemistry. So making self-plasticized materials, as was our goal. What about being able to make branch materials? Let's get back into making copolymers. Let's make some interesting different copolymers that we couldn't make by the prior chemistry. What about reacting, for example, with acyl bromides, bromo-acyl bromides, which are known as excellent macroinitiators, right? So again, the HBr that's present and the acyl bromides is sufficient to catalyze removal of the trityl group. Then reaction with that acyl bromide, you can put on a bromopropionyl group very efficiently. You can put on a bromo-isobutyryl group somewhat less efficiently, but you can get enough of a degree of substitution to make interesting comb copolymers. And we have studies underway to make those comb copolymers by using these things as macroinitiators. So I have run longer than I meant to, so I'm not going to talk about the hydrogel stuff, but I'll simply conclude by saying we have regiospecific synthesis of amylose-based block copolymers with PLA by this bromination, acylation, azidation, click step, 3-step sequence with very high yields, very high selectivity. We're able to control the density, the location, and the length of the branches. And by doing that, we were able to prove our hypothesis that we can make a great compatibilizer for PLA and starch acetate, which should be able to get us into some high performance biodegradable plastics. Separately, we were able to make these 6A, 2, 3B, so one ester type attached at 6, another at 2 and 3, cellulose esters with very high selectivity. And that includes making materials that are self-plasticized, so might not require small molecule plasticizers, and also macroinitiators for block copolymers. Oops. And I'd like to thank the people that did the work. I've had some great students, I've had some great collaborators, the people that funded this work without whom it couldn't have happened. And I'd like to thank you for your attention.
All right. Thank you very much for a fantastic presentation, Kevin. I've never really thought about starch as such a versatile polymer platform, and personally appreciated the carbohydrate synthesis walkthrough and the shout out to NBS Chemistry to modify C6. So changing gears slightly, I'll now introduce our second speaker for today, Dr. Camille Gadula, who's a professor of chemistry and biochemistry at UC San Diego and a co-director of the UCSD Glycobiology Research and Training Center. He's also the current executive secretary and program chair of the CARB division of the ACS. And Camille has won numerous awards over the year in his independent career, including the NIH New Innovator Award, the PIMSI Young Investigator Award, and has been named as both an Alfred P. Sloan Fellow as well as a Cottrell Fellow. Camille's research program at UCSD is aimed at developing chemical methods for engineering the composition and biophysical properties of the glycocalyx of living cells to control their interactions with biochemical cues, signaling, and differentiation. And for some background on Professor Gadula, he was born and raised in the Czech Republic, and he came to the US to pursue his graduate education in chemical synthesis, where he received the Alfred Bader Fellowship to pursue his PhD at Columbia University with Professor Dalibor Samesh. He then worked with Professor Carolyn Bertozzi at UC Berkeley and the Molecular Foundry at LBNL as a NIH K99 postdoctoral fellow. And then finally, Camille joined UC San Diego as an assistant professor in 2013, which is actually where I first met him back when I was a graduate student in the same department. So it is thus an honor to both share this virtual stage with him, as well as a distinct pleasure to introduce him to you today. So with that, take it away, Camille.
Great. Thank you, Cassie. Thanks for the very kind introduction. And thank you all for joining today. It is my pleasure to share some of the research that we do in our lab, and maybe from a different perspective of carbohydrates materials. Specifically, I would like to talk a little bit about how we use carbohydrates containing polymers as probes and tools to study the various biological roles of glycans at the cell surface. But perhaps what I should do first is to introduce glycans and what they do. And maybe I could do it by just posing a simple question. Maybe you have thought about it, but think about cells in an extreme environment, which could be just simply an air. Imagine what happens if you take cells out of their culture media and you expose them to air. What you will observe is probably cell death. The cells will rapidly die without being in water. As well, if you pour acid on cells in the laboratory, they will die. Or if you freeze them, again, the cells will form ice crystals and they will rupture and die. And so cells are not really well suited to survive in these environments, which we consider our natural environments, but to cells, they're extreme. So how do cells manage this challenge? We breathe air. We are able to use our lungs to exchange gases. We are able to digest food at very low pH, 2 to 4 pH. Fish are able to swim in sub-freezing temperatures in oceans, and they survive. So how does nature really address this challenge? And in fact, what's really interesting is that it has actually evolved a biopolymer to protect cells against these environments called mucins. And this will be the first subject of my talk today. So the mucins are really interesting large molecular weight polymers, oftentimes in the megadalton size, which don't really have the typical protein architecture with alpha helical and beta sheet folds that you might expect, but rather they look like this extended drawing of the mucin shown here. And the protein backbone, which is in the middle, is then decorated densely with complex, oftentimes branched carbohydrates or glycans, as we refer to them, which may look like something like that when I'm showing them here in the chemical structure. And so these carbohydrates do a few different things. They not only provide a hydrating environment around the protein backbone, but oftentimes they could also have charge as these terminal monosaccharide modifications shown in magenta called sialic acids. And so they could also have a buffering capacity, for instance, helping the stomach to maintain acidic pH without necessarily killing the epithelial cells. Another thing that these glycans do is that they actually force the protein into its unique conformation. So when you look at the mucin in more detail, what you will see is that that the polymers are organized into segments called variable tandem repeats, which are heavily glycosylated. And these glycans are then pushing the backbone into this sort of conformation that when you look at it under electron microscope or atomic form microscope, you will see it looks like a half-cooked spaghetti. And this is a very unique feature of this glycoprotein that contributes to the overall physical properties of mucins. You might be familiar with mucins as part of the mucus, which is the secreted mucus that covers, let's say, our GI tract and the upper airways. But mucins can also be anchored to the cell surface. And so here I'm showing you an image of an epithelial cell from the gut where the individual glycopolymers are projecting away, forming this very thick brush on the cell surface. And so what we are interested in our lab is to try to use polymer chemistry to understand how this brush architecture and composition affects biological processes that occur at the cell interface. So, but protection of the cell is not the only function that mucins perform. In fact, mucins are involved in a lot of other processes. They can coordinate interaction with the immune system, and they can present signatures of self that will tell immune systems whether cells are healthy or whether you might have a foreign cell, such a tumor cell in the body, to eliminate it. On the other hand, the mucins and the glycocalyx can also be exploited by pathogens and which can subvert the brush to gain entry to receptors on the host cell surface and infect the host. And so mucins are not only large biopolymers, but also they're biopolymers that encode biological information. We need to talk a little bit about how this is done. The dirty secret of glycobiology is that the interactions of individual monosaccharides or even oligosaccharides and the proteins that bind to them, which are oftentimes referred to as lectins, are relatively weak. So if you were to measure a binding interaction for binding of one lectin with one carbohydrate binding domain to a glycan on a protein, you will measure dissociation constant of about 100 micromolar, which is not very strong in a biological context. And so to compensate for that, nature uses what's called multivalency to increase the avidity of these interactions. And so you might have lectins that have two domains oligomerized together that can engage two glycans on a glycoprotein at the same time. And now the overall avidity of the interaction is the product of the microscopic affinities for each individual interaction event. And so because it's a product, you go from 100 micromolar to about 10 nanomolar. And that now is specific and strong interaction in a biological context. We are getting close to the type of interactions that you would see between antibodies and antigens, for instance. So this is called the face-to-face binding model, where you have multiple binding events occurring at the same time between the lectins and the mucin protein. But that's not the only one mechanism by which lectins can achieve multivalency and high avidity. There's another model, which is called the bind and slide model, where the lectin only binds to one glycan at a time, and then it can dissociate. But because of the local concentration of these other glycans along the polymer backbone, before the protein completely dissociates and leaves the complex, it can reassociate and rebind, and so it will continue interacting with the glycopolymer, producing overall high avidity event. Now, this also leaves one binding site on the adjacent lectin domain to cross-link to another glycoprotein on the cell surface. And so this allows for bringing glycoproteins together at the cell surface, and this is oftentimes involved in signaling events that may occur at the cell surface. And so I like to think of these two models, the face-to-face and the bind and slide model, as a digital model for the face-to-face, where you have one individual weak interaction and then very strong dual interaction, right? And so you go from 0 to 1, 0 to 1. Whereas the other model, the bind and slide model, is more of an analog model where you have a knob where you can turn the strength of the interaction by, for instance, introducing more copies of the glycan or extending the length of the polymer, right? Or increasing the density of these polymers at the cell surface. So these are really interesting two ways by which biology modulates interactions of proteins and glycans. So when we study these glycoproteins, we need to be able to access them in a very controlled way. And this is something that's challenging to do using traditional molecular biology tools, because glycosylation, which is the attachment of glycans to the protein, is not a templated process. So there's no gene that encodes a glycan structure or the time point at which it will be attached to a protein, right? So all of the glycosylation occurs after translation of the proteins. And so you cannot really use the DNA and the genome to template the production and synthesis of well-defined glycoproteins like mucins. Instead, chemists have long tried to explore polymers which look very similar in their overall architecture to mimic the structure of the proteins and also to introduce glycans that might be relevant to mucin functions. And so here are some examples of the polymer architectures that have been developed over the years. For instance, here on the left-hand side, you will see polymethyl vinyl ketone polymers prepared using Rath polymerization techniques, to which sugars could be introduced using oxyligation. More recently, these beautiful N-carboxy anhydride polymers developed by Jessica Kramer at the University of Utah, which now contain polypeptide backbone, to which glycans can be attached at the monomer stage. Or these wonderful ROM polymers developed by Laura Kiesling's lab at MIT, where the stereo control of the ROM process and the cis and trans isomerization can actually control the overall nanoscale conformation of the protein, and going from a mucin-like display of glycans to something that's looking more like a globular blob. And so there's a lot of interesting polymer strategies that one can use to build a functional model of mucins. In my lab, we are interested in using these types of materials to study biological phenomena. And so the first story I would like to tell you focuses on interactions of pathogens such as influenza A viruses with the mucin glycocalyx at the epithelial cell surface. So influenza virus A is a quintessential pathogen that uses sugars to get into host cells. And it has two proteins that engage human sugars on our cells. And the sugars that these proteins bind are these magenta terminal modifications called sialic acids. So the first protein that binds to these sialic acids is called hemagglutinin, and that's the H in H1N1, for instance. And depending on the configuration of the sialic acid, whether it's attached here to the underlying galactose, either alpha-2,3 position, those are viruses that would recognize that these are the avian type structures for viruses that are populating the avian reservoir. However, human cells produce mostly sialic acids attached to the alpha-2,6 linkage in the galactose. And so the avian viruses need to switch from binding to the alpha-2,3 red receptor to the blue green alpha-2,6 human receptor in order to move from birds to humans. And so this interaction is very important. The virus also has another protein called neuraminidase, which is an enzyme which actually breaks the bond, the glycosidic bond between the sialic acid and the underlying receptor. And this is the N in H1N1. And the viruses use neuraminidase to detach themselves from cells after they've been replicated in the host cell to infect others. And so, because this is an enzymatic process, you can inhibit it. And so in this image, you can see the surface of a cell that's producing new viruses, but it's been treated with Tamiflu, which is a neuraminidase inhibitor. And you can see that these viruses are attached and they can't leave. So this is the fundamental principle behind the function of Tamiflu. What my lab is interested in understanding is how does the mucin brush and the individual mucins actually affect the interactions of the virus? Do they provide protective functions, or do they actually help the virus to get into the cell? So to do that, we need an access to a model for mucin. And so we use, again, synthetic macromolecular scaffolds to which we can attach all these glycans that are relevant to interactions with viruses. To produce mucin mimics, where we can control the carbohydrate structure type, the density, the size of the polymer. And because these are synthetic materials, we can also introduce various different functional groups, such as anchors for printing on solid surfaces to generate a synthetic glycocalyx model in analytical platforms, or by simply introducing a lipid tail, we can insert these into the cell surface and actually build that brush that I showed you artificially on the cell surface. And we can have optical probes so we can track these materials in the cell environment. Okay, my lab does not want to do much carbohydrate chemistry, which is very complicated, as we heard in the previous talk. And so instead, we try to develop a synthesis strategy that will allow us to just take glycans from natural sources, which oftentimes contain reducing ends, which are masked aldehydes, and then functionalize the polymer with an alpha heteroatom nucleophile to simply promote formation of a stable amino oxy bond between the glycan and the polymer. And so that's shown here. And so this will be our general strategy for bringing these really complex glycans into the fold of the polymer. Okay, to do that, in reality, we generated a library of polyacrylamide polymers, most ranging in size from approximately 35 to up to 600 or so repeating units that have the functional amino oxy side chain, a fluorophore on one end, and an anchoring unit on the other. And then we can simply incubate these polymer backbones with our sugars of interest under mildly acidic conditions, dialyze away unreacted sugar, and collect our purified material, which we can characterize by NMR. So this actually brings me to an interesting question. Just out of curiosity, I would like to pose a question to you. When we think about these glycans, how many building blocks are we really talking about? So for instance, if you think of DNA, my question to you is, how many nucleotides are there that form the complete assembly of DNA structures in the human genome? What are the amino acids that encode, or how many amino acids are there that encode the human proteome? And finally, if you could tell me, how many monosaccharides are there that encode the human glycome?
And I've just activated a poll question, so you can let us know. It should appear for all of you. A reminder, we are also eager for your questions for Camille, so feel free to enter those into the Q&A window at any point. And seeing some results start to come in, we've actually never tried this type of poll question before, so curious to see how the results look. All right, I'm seeing the responses coming now, and I'm going to close this poll in 5, 4, 3, 2, 1. And looks like the details are hard to view. There's a wide range of numbers, but Camille, I'll turn it back over to you to let us know what the correct responses are.
So I can't really see the specific responses, but my guess is that you correctly identified the number of nucleotides that form the DNA, which would be 4, and the 20 amino acids that are the canonical amino acids in human proteins. However, what's much less known is that there's actually a very limited number of monosaccharides that form the human glycome. And in fact, the general number that's accepted is about 9. And I'm showing them here as represented as individual structure, as these little charms, which represent the unique chemical structure of the sugar. So we have glucose, galactose, and acetamide sugars, as well as, let's say, fucose and xylose as a red triangle and orange star. And here, represented as a magenta diamond, is our sialic acid, also sometimes referred to as nu5AC. And so the repertoire of building blocks is really not that great, but the way that they can be connected together creates a lot of structural space. And so here's just a subset of of glycans that we were able to introduce into our polymer, shown here, where we are combining these different building blocks in different configurations. And to give you a sense of how efficient this chemical conjugation strategy is, here's an example of a hexasaccharide, which is branch, and it also has the negatively charged sialic acid and is a magenta diamond. And under the conditions that we use, we typically get about 45% side chain substitution on the polymer. So about every other side chain would carry this large glycan. And this packing of these glycans then gives the polymer the characteristic extended mucin architecture. So how do we use them to actually study viral interactions? So what we could do is we can now generate a library of mucin mimetics, which have both the avian and human receptor structure in red and blue. And then we can vary the number copy of these glycans on the polymers, but also the polymer size. And then finally, we can print them in microarray format to control the brush density on the surface. And then we use AI to try to discern whether there are specific patterns that the virus would recognize and potentially distinguish between the human and bird viruses, you can see here. But first, what we wanted to know was, how does the presentation of these individual receptors affect their interactions with the virus? So my student, Taryn Lucas, who's now a postdoctoral scholar at Yale in Stacy Malaker's lab, first synthesized mucin mimetics, which had either the red avian receptor or the the blue human receptor type. And then she varied the length of the mucin mimetic glycopolymer from about 150 repeating units to about degree of polymerization of 460. Okay. And then we measured the strength of their interactions with the viruses. And for that, we could actually use red blood cell agglutination assay, where the red blood cells, because they carry the receptors for the viruses, can be agglutinated and clustered together by the virus. And then we measure the inhibition capacity of our mucin mimetics to stop that agglutination process from occurring. Okay, so first, what Taryn did is she just looked at the individual trisaccharides before we even put them on the polymer scaffold and see how strongly they interact with the virus. And you can see that that both of these structures, the blue and the red human one and the avian one, inhibit red blood cell agglutination with about 50 millimolar or 12.5 millimolar inhibition capacity. So this is very consistent with what I told you, that the individual interactions of glycans and proteins are quite weak, right? We're in the millimolar range. So here's my second question for you today, to see whether we can apply the concept that we learned a few slides back. And can you help me predict what sort of binding range or strength of interaction should I anticipate for mucin mimetics with degree of polymerization of 460, so the longest possible mucin mimetic that we can make? What sort of binding range would you think you would observe that would allow you to inhibit the agglutination of these red blood cells with the virus?
And you should see the poll question that has just popped up. Your choices are about 1 millimolar, about 100 micromolar, about 1 micromolar, about 100 nanomolar, or about 1 nanomolar. And I can see about getting close to 30% of you all have voted. So I'm going to be closing this poll in 5, 4, 3, 2, 1. And the top results, both C and D, were tied. So with that, Camille, I'll turn it back over to you.
Yeah, excellent. So you've been paying attention. We talked about the different mechanisms through which avidity can be enhanced through multivalency. And in fact, what we observed in our materials was that when we increased the length, or when we created the short polymer mimetic with about 70 copies of the trisaccharide, we did see strengthening of the interaction to about 600 nanomolar for the avian glycan. And as we go to the longest one, we go to about 78 nanomolar. So 100 nanomolar, about D was correct, but anywhere between C, D, and E, you would have been correct in your prediction. But what you observe here, which is really interesting, is that that's not the case for the human type receptor. We only observe this behavior for the avian one. For some reason, the virus, the H1N1 virus, does not like interacting with the human receptors when they're presented in the mucin conformation, which sort of brings us to potentially an explanation for how is it possible that our epithelial cells that are attacked by the viruses can actually present this very dense brush with millions and billions of copies of the receptors without actually making it easier for the virus to enter the cell. Next, what we could do is we can build the glycocalyx brush on the surface and see how the brush density would actually affect interactions. And so here we are printing our polymers with different receptors, increasing size and valencies, and then increasing concentrations to really modulate the different parameters of the brush. We use the fluorophore in the polymers to tell us how much we're printing, and now we're looking for binding of the virus to the surface. And what we observed was really quite interesting. In all cases, as we plot the overall density of the receptor for the virus in the spot, the virus binding goes down. And then we looked at it a little bit more closely for the red avian receptors and the humans, and we can see that as the polymers get taller and as the glycocalyx gets denser, we actually get less binding of the virus overall. And this is more pronounced for the human type structure. So not only the mucin architecture, but also potentially its crowding in the glycocalyx brush can contribute to protection of the cell, despite the fact that we are really packing a lot of these receptors for the virus in the glycocalyx. Okay, so we're starting to maybe think of some mechanisms for how the mucin glycocalyx may interact with the virus, and we think that it's providing protection by basically creating this thick brush that the virus doesn't really necessarily like to bind to very well. The taller and denser the less it likes to interact with it. But we also know that the virus has a neuraminidase that can cleave these sugars, these magenta diamonds, and potentially get through the brush. On the other hand, the brush can also counteract the virus by shedding from the cell surface. And mucin-induced shedding is a common mechanism for limiting viral infection in the airways. But the question for us was, okay, fine. The virus can cleave these sialic acid, but the brush is still there. So how does it get through that really thick brush that I showed you at the very first, in the very first slide? And so for that, we wanted to generate it chemically. And so we designed a new synthetic polymer backbone, which is based on polyethylene glycol, again, made by controlled polymerization techniques. And now instead of these sialic acid receptors, we're putting glucose, which is a monosaccharide that the virus doesn't like to interact with. And then we designed the polymer so that crowding along the backbone would then produce this sort of mucin type architecture. And so what you could see here is a polymer decorated with a disaccharide lactose, which is about 40 nanometers in size. And so that's degree of polymerization 400. We also generated some much shorter versions of this polymer. And the reason for that is because we wanted to now use them to build the mucin glycocalyx brush directly on the red blood cell surface. Remember, the red blood cells have the receptor for the virus, but if you look at the glycocalyx, the natural glycocalyx of red blood cells is about 10 nanometers in size. And so we can start building our mucin mimetic brush without the viral receptors, and we can make it shorter than the natural glycocalyx, or we can make it longer. And we can also control the density of the brush. And then my student, Dan, measured how quickly the virus would bind to these cells in the presence of the brush. And what we see is that as we increase the size of and the dimensions of the glycocalyx, the binding becomes slower, which makes sense. It's consistent with sort of shielding of these underlying receptors as the brush grows taller. And that could be what we would understand as the function of the natural glycocalyx, sort of shielding the cell surface. However, Dan also did another experiment where he let the system equilibrate, and then we measured how quickly the virus will dissociate from the cell surface. And he observed something that was unexpected. He found that all polymers, regardless of size, actually enhanced retention of the virus. The virus, for some reason, bound stronger to its receptors in the presence of the crowded brush. And so that's something that we wanted to look at because we wanted to understand how this functions. And so we used a model lectin instead of the virus to do microscopy. And we have our red blood cells, and they're modified with our green polymers. And then we have a red labeled lectin that mimics the hemagglutinin of the viruses. And what we see is that they don't really occupy the same space in the membrane. And so we started to think whether there's some sort of crowding event that may promote clustering of these models for the virus. And so we designed a FRET system where we could measure the distance of these two lectins decorated with donor and acceptor FRET pair in the presence of the crowding glycocalyx. And what we saw is that when we crowd the glycocalyx, the interaction between those two lectins increases and they get closer together. So the brush is forcing them to cluster together. We did the opposite experiment where we look at the crowding in the glycopolymer brush. And again, the lectins were able to push the glycopolymers closer together, clustering those together. And that explains how the green and red colors phase separate in the membrane. And that led us to propose this mechanism for how the glycocalyx can actually be functioning at the cell surface. So because of its extended size and density, it shields the cells from the virus. And so it slows down binding of the virus. But as the virus starts to bind, it can use the crowding in the brush to promote its interactions with the receptors. And you can imagine that if the brush is getting crowded, some of the smaller components will be diffusing into the void under the virus. And oftentimes these smaller components could be small glycoproteins or glycolipids that have the receptor for the virus. And we're increasing the multivalency and the avidity of the interaction. So this is another example of how biology could use multivalent display of receptors to promote avidity of interactions in the system. And so this is something that we're now investigating in more detail. Okay, in the last couple of minutes, I would like to touch up on another aspect of the glycocalyx that we're very much interested in the laboratory. So here I'm showing you the glycocalyx that would be found on the lining of blood vessels. For instance, here, that's the endothelium. And here, there are other types of glycoconjugates other than mucins, such as shown here, where the proteins are decorated with long polysaccharides that promote interactions with signaling proteins, and they can actually regulate how cells sense the outside environment. This is particularly interesting and important in the context of growth factor signaling that we study in our lab. So the growth factors that promote cellular signaling and, for instance, differentiation of stem cells, which is one of the big areas of our research, require these polysaccharides for function. So in mouse embryonic stem cells, these polysaccharides recruit growth factors and promote organization of signaling complexes that then starts the signaling process and eventually leads to differentiation, for instance, neural differentiation. If you delete those polysaccharides, the cells cannot sense the growth factor, the complexes don't form, and you don't get differentiation. And so we thought maybe you can borrow pieces of the long polysaccharide, reorganize it on the polymeric scaffold, introduce it to the cell surface, and reignite or reinitiate these signaling pathways and actually guide the cells to differentiate it into a direction that we want. The glycans, the polysaccharides, are very complex, but they do have these very highly sulfated polyanionic regions, which interact with positively charged amino acid residues on growth factors and their receptors. And you can see one shown here. We didn't want to make them again, but we knew how to break these polysaccharides by using enzymes that cleave the glycosidic bonds in the polysaccharide. And it breaks down the long polysaccharides into disaccharide repeating units of the polymer, which still contain the sulfate patterns in them. And the sulfate patterns are believed to encode the specificity of the growth factors and their receptors that we want to exploit. And again, we have reducing ends in these disaccharides, and we can introduce them into our polymeric scaffold that I showed you before. And we can now print our polymers on a microarray, and each of the spots corresponds to a polymer with a unique sulfation motif on the disaccharide. And then we probe them for interaction with growth factors of interest, such as fibroblast growth factor 2 for neural differentiation. And you can see that we get some winners that engage the growth factor quite well and some that don't. Okay, so now that we have polymers that can bind to our growth factor of interest, we can introduce them to the cell surface of the genetically modified cells that don't produce the polysaccharides of their own. And we do it by simply attaching a lipid anchor to the polymer. And like dissolves like, and the polymers anchor themselves into the lipid membrane of these cells. And you can see, because we have a fluorophore, you can see these polymers on the surface of these embryonic stem cells. And now that when we stimulate the cells with the growth factor FGF2, we see a reactivation of the signaling pathway. And after 6 days, we see the cells to undergo neural differentiation towards neural rosettes. But however, the ones that didn't have the polymer or had polymer that didn't have the disaccharide that binds to the growth factor still remain undifferentiated in this state. These materials are synthetic, and they're great for tissue engineering applications, and you can imagine that we can use them. And in fact, we can actually use the size control in the polymer architecture to control how quickly these polymers diffuse into three-dimensional multicellular objects such as embryoid bodies. And you can see that short polymers will diffuse very rapidly into the embryoid body and promote interaction of FGF2 all over the spheroid. However, the much larger polymers diffuse a little more slowly, and so you only restrict the growth factors only to the periphery of the embryoid body. And that's something that the natural polysaccharides do in tissues. They pattern interactions of growth factors and development of organs and more complex organisms. We could also integrate them into hydrogels and seed cells directly over these materials and study how the materials promote or inhibit interactions with growth factors and promote signaling in stem cell arrays. So these are some cool applications of these types of materials. Finally, as we were trying to think about maybe using the control of interactions of stem cells within their environment for biomedical application, perhaps stem cell-based tissue repair, we need to figure out how to target these materials to the stem cells that we want to differentiate after we introduce them into the body. The lipid approach is cute, it's nice, but all cells have lipid membranes, and so we need something a little bit more specific. And for that purpose, we take advantage of proteins that are unique to stem cells that you will not find in other already differentiated cells and tissue. And those are oftentimes referred to as embryonic markers. And we could use targeting elements such as DNA aptamers conjugated to our polymers to target the conjugates directly to the stem cells and promote interaction of the growth factor there without really necessarily affecting the others. And so you can see the cells that don't have their natural polysaccharides, when they're treated with the DNA aptamer conjugate that has the right sequence and binds to the embryonic marker, after 6 days, we start to see neural cells to start to differentiate. However, cells that are targeted with conjugates where we scramble the sequence of the aptamer, it's not targeting the embryonic marker here in magenta, the cells remain still mostly undifferentiated and purple. So now we actually have a good way to target specific cell types depending on what sort of proteins they express at the differentiation stage. So we're starting to think how we might also inhibit growth factor interactions, and this is a new paper that will be coming out soon, which we're submitting, where we can take advantage of these interactions between the growth factor and polymers and receptors that recycle between the lysosome and the cell surface and actually drive degradation of growth factors in cells, for instance, cancer cells, to inhibit their growth and differentiation in patients. Okay, so with that, I'm out of time. But what I would like to just sort of summarize is some of the key take-home points that I would like to reinforce through my talk. Hopefully, you learned some interesting aspects of glycans and their roles in regulating cellular interactions, be it with pathogens or with signaling proteins, and that synthetic polymers really offer a unique architecture, a unique tool to model these glycoproteins that we would see on cell surfaces and use them as tools to probe or even control the biological processes that you are interested in. Control polymerization techniques are obviously important because of the multivalency mechanisms for achieving high avidity. And then I already touched upon the fact that you can then engineer these materials with unique glycan structures to target specific glycan-mediated cellular processes for biomedical applications. With that, I need to thank my team and collaborators at UCSD and that did the work. And also, I would like to sort of give a highlight to our Glycobiology Research and Training Center at UCSD, which creates a really unique environment to study glycans. And so if you're interested, please reach out to us. If you have any interest in doing glycan research, we are here to help you with a range of resources and educational activities. And with that, I would like to answer any questions that you might have.
All right. Thank you, Camille, for a very wonderful talk. So now we have a little bit of time. I've been tracking the audience questions. So